Gentlemen, and welcome to this session of Global Health Conversation Series. This conversation series is a way to continue engaging with our international partners in a meaningful way, which allows us to share and learn from best practices worldwide. It is an honor for me today to lead this conversation, and my name is Francis Tungia, and I'm Associate Professor at the Center for Global Health and Infectious Diseases at the College of Public Health, University of South Florida. My research is on host parasite interactions and vaccine development for malaria, with a focus on Plasmodium vivax malaria. So today we will be talking about malaria, which is often referred to as one of the neglected tropical diseases. And it's a disease that kills hundreds of thousands of people yearly, incapacitates many more, and it causes a great economic toll on the endemic regions, mostly these countries lack affordable healthcare, and yet it is not given the much needed attention that it deserves. So today, one of our international collaborators and a dynamic research scientist from Brazil, Dr. Luzia Carvalho, will be presenting her work and some of the research that we've been doing together for the past more than 8 years. So Dr. Luzia Carvalho is a principal investigator of molecular biology and immunology of malaria at the unit at the René Rachu Institute, Fiocruz, Minas, in Belo Horizonte in Brazil. Dr. Carvalho's group has devoted a great effort to characterize the protective immune responses against lead blood stage vaccine candidates in populations living in the rainforests of the Brazilian Amazon. So we have been working together for more than 8 years today, and it has been a very productive collaboration, not only in terms of publications, but also in exchange of students and research scholars between our research teams and institutions. Before I continue, I would like to acknowledge the presence of Professor John H. Adams, who is the backbone of this collaboration between our research teams, and he has been my mentor for many years. So today, we will have the opportunity to hear from Dr. Luzia Carvalho, and we'll also have the opportunity to ask questions with regards to her presentation and other areas related to malaria and global health. So I would like to, at this juncture, to hand over the microphone to Dr. Carvalho, who will be presenting his talk on the 10-year follow-up study of Plasmodium vivax Dophi binding protein region 2 immune responses in the Brazilian Amazon. Let's welcome Dr. Carvalho.
Thank you very much, Franz, for this very nice introduction. I will share my screen. You can see my screen?
Yes.
Okay, thank you very much. Thanks, Franz, for a very nice and sweet introduction. First of all, I would like to thank the organizer for the opportunity to share our results here today. That's part of a long-term collaboration with John Adams' group at the Center of Global Health and Infectious Disease Research. Before I talk about my results, I'd like to share some key information about my institute in Brazil, the Oswaldo Cruz Foundation, that's Fiocruz, that's part of the Brazilian Ministry of Health. So Fiocruz is the most prominent institution in Latin America in science and technology in health, and as stated here, produce, disseminate, and share scientific and technological knowledge to promote health and social development. This is summarized here, and also aims to intensify research in health. Fiocruz becomes now articulation hub for a global health network in Latin America, and also part of doing all this generation of knowledge, education. Fiocruz is very important for our public immunization of the population. We have this our public health system in Brazil that's called SUS, S-U-S, So Fiocruz produce a vaccine, for example, for the public health, for the SUS. For example, the yellow fever vaccine. Also, Fiocruz produce medicine free for the public health system, include for malaria, for also chronic disease like diabetes. Also have a very strong role in Latin American surveillance, include the genome of virus and now SARS-CoV-2. Fiocruz also has a hospital clinical care for infection disease, reference in clinical infection disease. That's in our main office in Brazil, that's in Rio, where you can see this beautiful castle. Fiocruz is in different states of Brazil. The unit that I'm linked is Fiocruz Minas, that's here in Minas Gerais. But also we have a collaboration, and all these units of Fiocruz, we are connecting a network. For example, I will present here today results of our study in the Amazons, make with collaboration in the Amazons, also results for making the Acre, where malaria is endemic. So my institute here, Fiocruz Minas in Belo Horizonte, we have two postgrad courses, one in public health and the other in health and science. We have a lot of facilities, for example, include flow cytometry, bioinformatics, bioassays, next generation sequence, etc. We also have a reference center for neglect disease, include Chagas disease, leishmaniasis, and other diseases. Also have biological collection. Probably we have the biggest collection of phlebotomine sandfly, that's vector of leishmaniasis in Latin America. And all these research and these students here, we work in different fields of investigation. We have a strong background in infection and parasitic disease, mainly to work in the new diagnostic assays for tropical disease, for neglect tropical disease, and also in vaccine, immune response, pathology, etc. And also in arbovirus, now strong in arbovirus. And Fiocruz also have a tradition in the field of social science. We have very strong groups in epidemiology. For example, do study age and health, and now also people working in environment and health. So we have different fields of investigation. If someone would like more details about this, I may give here the contact of our international support team. This is from Fiocruz Minas. Picture of our city, about 2 million people. France, you must come here. So, Isame Franco, you can contact us. If someone would like to know more about Fiocruz, you can go to their website. We have this in English and in Spanish also. So, back to our study. What I'm present here today is about our study of Plasmodium vivax. This is the more spread Plasmodium malaria parasite in the world, and the immune response against DBP, the Duffy-binding protein. And why this protein is so important for vaccine as vaccine candidate. For the people that are not familiar with malaria, this parasite Plasmodium vivax, most of the Plasmodium vivax strain depend on the interaction between the Duffy-binding proteins, in special the region 2, that's the ligand domain that bind to a receptor, the receptor Duffy, blood group, or also known as DARK, because a chemokine receptor, to interact with the reticulocyte. So most of Plasmodium vivax strain depends on this interaction to get blood infection. So this makes this a very relevant candidate for vaccine. And this also explains why Plasmodium vivax does not predominate in Africa, where most of the people here are DAF negative. This was a receptor described by Lou Miller about 15 years ago. So you can see that Plasmodium vivax predominate here in Latin America, in Asia, especially in South of Asia, and some parts of Africa also. So what do you know about the antibody response of DBP in humans? We know that people exposed to Plasmodium vivax develop this conventional antibody response. For example, here in the Amazon, the probability of having DBP antibodies increased 2% every year of exposure. But what's important that part of this antibody response include those antibodies that are able to block the interaction between the DBP2 and the DAC receptor, include here in the Amazon area. So these antibodies are able to block this complex interaction between the DBP and the DAC that make these tetramers. So, and also what's important for vaccine development is that these inhibitory antibodies, they are associated with some degree of clinical protection in the field, including here in Brazil. But what's bad news about this is that very low proportion of Plasmodium vivax exposed individuals acquire this strand-transcending inhibitory antibody response. Antibodies able to block this interaction between different DBP strains. And this is not only in Brazil, for example, that we have a malaria that's equal to meso-endemic, but also in highly endemic areas, for example, such Papua New Guinea. So, and I think that one of our good collaboration with John Adams that start was like this, probably will make the more convinced, will provide the more convinced evidence that DBP immune response is allele specific. So how we make this in Brazil, the endemic region of malaria is here, the Amazon in green. And what happened that we have an outbreak of malaria. So these people here in my hometown, where Fiocruz is located, they were naive people that having been exposed for the first time to malaria, to Plasmodium virus. During this outbreak was a fisherman that came to this area. Here we have the vector, where we don't have the Anopheles darlichia, we don't have the sick people. So a fisherman arrived here and we have a small outbreak. So people was first exposed to Plasmodium virus. So what was our approach, experimental approach? In this area, we identify a single DBP allele during the people, it's about 20 cases. So we have this a single allele during the first infection and relapse also during the relapse. Plasmodium vivax are relapse disease. They can get new infection because we have these dormant latent forms in the liver. So we express the DBP from the outbreak in this surface of core cells here. Transfect express this DBP from the outbreak because Plasmodium vivax don't have long-term culture of blood stage like falciparum. And when you can see here, when you incubate the sera, the human sera from the outbreak with these cells express the DBP from the outbreak, you can see the IgG, this anti-IgG in red, human anti-IgG in red. You can see that sera from the people from the outbreak just bind to the homologous allele. Cannot bind, for example, to SAL1 strain of Plasmodium vivax. This is a reference strain of Plasmodium vivax. You can see they cannot bind. So show to us that, yes, this is strain specific, this antibody. Just illustration, these inhibitory antibodies, very low titers because this is prime infection. You can see in blue, a titration that is the three people here, 3 individuals. You can see that they bind to the homologous, but not to the heterologous. So yes, this was a great contribution, so that without doubt that the immune response against this is strain specific. And this can be explained by this huge polymorphism of DBP in the different endemic areas. This was made by Thais Souza, that was our postdoc, and now she's a researcher of our group. And what can see, every single color is a different allele of DBP. SAL1 here is in blue, for example, predominate in Papua New Guinea, but not here in Brazil, not in Colombia. So what we can conclude about this, that yes, the DBP immune response is a protein that is essential for the parasite, but it's weakly immunogenic, induces strain specific. But the good news about vaccine is that few people, including here in Brazil or there in Asia, they develop this strain transient immunity. There are very few people, but they have this inhibitory antibody response against different strain of Plasmodium virus. So we call these people elite responders. So this is the people that encourage that DBP should be a good target for vaccine development. So this means that DBP includes conserved immunogenic epitopes that can be targeted for a more broadly inhibitory antibody response. So this is the idea of John Adams, that it's possible to design a DBP immunogen that target the immune response for relevant and conserved B cell epitope. So the idea of Decno family was this, to bypass this polymorphism of DBP. So in the first of this construction of Decno, so what Francis make, this was a pioneer work of Francis and John. So what Francis make this, the construction was delete what's read here is the DEC, the more polymorphic epitope of DBP. So what Francis showed that this worked very well in mice. So yes, this Decno could induce a broadly inhibitory antibody response in mice against different DBP variants. But when we check this Decno, if the people here are elite respond in the Amazon, if they can recognize this Decno too, no, they could not. So nobody's perfect, right? So come to the second generation of this Decno family. So Camila Peter was my former postdoc that went to John's lab and worked with Francis there. So what they make, they improve this Decno 2. So this all in red was a lot of polymorphic residuals change in the Decno 2. And yes, what they show here that now the people that the Amazon that you call the elite responder, yes, they have high titer of Decno 2 antibodies. And this was very promised, and they have a huge titers, and this was associated with that elite response. What also was good that France came to Brazil, and also with Flora, they make short-term plasmodium vivax culture here in the Amazon area. As I told you before, plasmodium vivax only have short period of times of blood stage culture. So Francis teach us how to make this reticulocytes invasion assay. So what is shown here in this here is that antibodies against Decno 2 is able to block the invasion of the merozoite into reticulocytes. So yes, we have a very good construction that people from the Amazon recognize Decno 2. These antibodies are able to block the invasion of the parasite. So the next question that I would like to check, I started to show you that you have this outbreak here many years ago, close to our lab. So the question that you make that is very important for a vaccine is that the people that have made a single episode of malaria infection about 13 years ago, they still have memory B cells against this malaria. So we check, this is just to illustrate, to make a follow-up of that people baseline 3, 6, and 1 year later. So this is conventional IgG antibodies against Decno 2 because we have the frozen sera. You can see here that you have sera for people has the antibodies against this, increase here because the time they have relapse, not a new infection for the mosquitoes, but a new blood infection because the parasite was epinozoites wake up there. So, but 30 years later, they don't have antibodies circulate. You could not see this not only for DBP, but also, for example, for much more immunogenic plasmodium vivax proteins such MSP190T, AM1. So yes, they don't have circulate antibody. So what we check, this is Barbara, this was the PhD students of lab. What Barbara make was a B cell spot. So here is plasma cell per million. They are specific for Decno 2. This we check this in case, case was the people at that time they have blood infection and symptoms, and no case they don't have a positive blood smear and they don't have symptoms. So we check this case and no case. Can see very few people that could localize, but they are single infection people. And also include people non-exposed, that people that move to this area here after the outbreak. So you can see that, yes, they have not so much, but they have a specific B cell response, plasma cell specific for Decno 2, means that they have memory B cells. Here, just to illustrate this very preliminary data. So we decided to check the different gene expression, immune gene expression from the case and no case. Yes, you can see there is difference in response between this and this. That suggests they all hypothesis correct. Yes, these people, they have different types of memory. Because this is very few cells, what we're trying to do now is start to sort these cells, to purify these B cells, because in the peripheral blood of human, you have very few precursors of B cells. So what we have to do is the protocol for Silat, was a former Jones lab postdoc, and she make a tetramer, she biotinylate the DECNO2 and make a tetramer with streptavidin. So the idea of this tetramer is to purify these B cells, concentrate these specific B cells for DECNO. Sai make this in mice, was worked very well. So I try to do use her protocol now for human here. So go to the field. Most we have been doing this long-term follow-up in the Amazon area. It's about a small community, about 2 hours from Manaus, and Floracan, that's a research from my group in Brazil that coordinate all this field work. So we have been follow about 500 people. Just to illustrate for you guys how is this area, we have people. This we have to use some kind of boat like this. We have this that is small streams that people live around. So we have to take this and usually go 3 to 4 hours to arrive people like this. That's called riverine population here. But I also have people that live close to the unpaved road that is less exposed to the mosquito bites. This is our house from Fiocruz there. Just to know you, this area we have leishmaniasis, we have different type of virus, for example, Mayara virus, Oropush virus, that's not very well studied. So we have a lot of potential virus to study in this area, and also leishmaniasis. Leishmaniasis is very common in Brazil, in the Amazon area. So yes, we have can work here. And this is the population for our study area. You can see by the phenotype they are native from the Amazon area. Yes, when you check the ancestry of these people, these were done by Thais Souza for our group. So in Brazil, we are like this. We are mixed between European, Africa, and Amerindian. Nobody here have one, just have a composition of this admixture of this. So in this area, we have much more Amerindia background. You can see that our study population about 4% of Amerindia. Also in this area, check dark genotype, because you know dark is important for the type of antibodies that these people produce. And also there is difference between the susceptibility, it depends of your dark genotype. In this area, you don't have the dark negative that's more common in Africa. That's more people that are more resistant to plasmodium vivax infection, because we don't have here a strong Africa background. We have here Amerindia background. That's not dark negative. So in the area where you have more African background in Brazil, there is no malaria. So in this area, it's more important, the Amerindia background. So I can see here, for example, that when you increase the ancestry of the Amerindia here, increase here, we decrease, for example, the allele B, that's in green, that's more common in European people, but we increase the allele A, that's more common in Amerindia, in Latin American Amerindia population. And also so in this area, what people have been shown before, that in this area also that allele B is more associated with more susceptibility for clinical malaria, and the allele A is more associated with more protection against clinical malaria. So we'd like to see also if what make a good and a bad inhibitory antibody response in this area. So we check a lot of group family of HLA class 2 that's more associated with antibody response. So we can see here, we have a lot of help here for Douglas Pires, that was from Fiocruz, now University of Melbourne, and David Asher. They have been work a lot with in silico. And what we demonstrate that difference in the peptide bind growth can explain difference between good and can partially explain the difference between good and bad antibody response, including those that inhibitory antibody response. I'm not going to this detail, just to show you that yes, there is some allele groups that's associated with a good and bad antibody response. And this is not specific for malaria, because if you check in the literature, you can see, for example, that one that's good for produce neutralized antibodies against DECNO2, against DVP, is also, for example, the people that has a good response for conventional vaccine against virus, for example, hepatitis B, influenza, etc. So this was very interesting. Back to the field, what we have been doing during all these years, we have to stop, if everybody knows, the pandemic virus. There is a time that in Brazil, the pandemic virus, the epicenter of in the Amazon state, we have a very complicated situation there. And still now with the people, the native people. Yes, in blue is the Vivax transmission. When we start using 2008, we have still falciparum in our study area. Now we don't have more, it's Vivax. You can see here, for example, there are years that there is no very low malaria transmission, could not detect a good infection in our study population. So this very long cohort allowed us to identify those people that you say that's a little response, these people that are this persistent antibody response, neutralized antibody response. Here, just to illustrate it, the conventional, this is the persistent, this people that has this group here, the PR is persistent responder with neutralized antibodies. Just to illustrate how is the conventional antibody response of these people, called phase 1, phase 2, when you don't have in that years without transmission, is the same. They don't decrease antibody response. In blue is the response against SAL1 that also is common in this endemic area here, in this our study area. And we say that SAL1 is more associated with the strain specific. And the response of DECNO2, that's it's not derived, it's not towards the specific epitope. So we say that's more transcending. So because it's conservative B cells epitope. So if you look like this, you say, okay, this is not people that do not have the inhibitory antibodies, can see that they don't respond so much. But if you check this here, you say, okay, they have the same response for SAL1 and DECNO2. They have the same response for strain specific and not strain specific. But in fact, this is not true. If you go here in more detail, this was the thesis of my student Camila Medeiros, that she's here. And what we saw here, this is the ratio between IgG and IgM. IgG is in white, IgM in gray, and the strain specific in blue, that's SAL1, and DECNO2 in red. You can see here during the first year, for example, you cannot see difference, nothing between nobody. But when you go to this many years later, this low transmission phase, you can see that you start to see that the white predominate the red, that's IgG for DECNO, and in the gray, it stay more blue. That's for strain specific, SAL1, and remain like this during the year. So you can split that IgG all the time. I think it's easy to check here in this pie. You can see that IgG towards start 50%, and during the years, start most of these people that has this persistent antibody response, they towards the antibody response to DECNO2. And what was, so this believe that's more associated to this acquired protection. So this IgM, what you can see here that you have, they don't increase, these people, they don't increase, IgM is very low against DECNO2, but remain the same for the SAL1. What's the strange? Oh my God, so long this IgM response. So we don't know what this IgM is doing here, but perhaps what we are saw is something that has been changed more recent about malaria. They believe that IgM in malaria can have a more relevant role that was before. So there is a paper that this review say that IgM in malaria could be faster, longer and stronger. So what you learn in the books was like this. And yes, if I go to the outbreak that I showed you before, we saw exactly like this picture of book. So if I'm go check the response of DBP, for example, in the outbreak, like this. The people got a strong IgM response. When they have a relapse, they boosted this IgM response, include for DEC-02. But IgM is like this. A bad antibodies that's not boosted. But now what they are saw not only for malaria, most of the results are for mice and also for flu. It's like this. We have this IgM that at least in mice, they have hypermutation IgM, not only IgG. They are strong IgM and specific IgM response that can be boosted and stay like this. There is a paper from Falcipro that I include here, but they follow people from returning to Australia after being exposed to Plasmodium falciparum, and this was antibodies against the whole merozoite blood stage of Plasmodium falciparum. They followed for about 6 months, these people, and they saw something like this. So what we believe that we are, so I don't think there is no long follow-up like ours in Vivax or in falciparum that many years. So what we believe that we are saw in our endemic area is something like this. That's very interesting, and I should open question that we should to investigate how is this B cell. What's this IgM is doing that? Perhaps this IgM is there because they can be stimulate, or the plasma cell can be stimulated, or B cells can be stimulated for the parasites. We know that the plasmodium virus can remain in the spleen, in the bone marrow for many years. Who knows this biomass of the parasite can be responsible for this, so we don't know. There is people that believe that the IgM is important to keep the IgG response. In this area, when IgG increase, the IgM decrease, so we don't have simultaneous both. But this, I think, depends on the protein used. So just to illustrate this. Back to the field. Oh, there is something that's close me here. What we are doing next. So we, after this worst part of the pandemic, we are returned to the endemic area. So in this year now, 2023, 2022, we are going, Flora and we are going again with our students and collaborates to the endemic area. So we are planning a new trip in July. So the idea now is to check what happened. If they still have, they can see here, this was the data for 2020. You can see here, they still have this elite responder there. There are not so many people, but yes, they are still there. If you check the peripheral blood of these people, ex vivo B cell, you cannot see this is elite responder, this, for example, is persistent non-responder and transient responder. We cannot see difference in the ex vivo, and this is not surprise because this is not a good infection. Just check my time. Okay, this is not a good infection, so we did not expect that we could see difference between these ex vivo B cells in the peripheral blood, because think this people has a lot of virus infection or other infection. So yes, they are not a good malaria infection, so no difference in the ex vivo. But if check, for example, this IgG plasma cell, IgG specific per million, you can see, yes, they need to respond, they have specific plasma cells for SAL1, that's common in that area. They have it also for DEC-02. I don't know, he is only IgG. I think there is something wrong here because this is supposed to be IgM. Yes, they only not have only IgG specific, but IgM, I don't know what this was. Probably this is IgM. I think my slide's wrong. But just to know, for you know that yes, they have it. This also is a differential gene expression. I'm not to go to this. Just make like this to check if there is difference. This has been done for Samik that's at that time was master student, and Samik now is a PhD start, have PhD student, so we go more deep in this. So just to show, yes, there is difference between the elite responder and no responder, and there is difference between also what's up and down regulate immune genes for DEC-02 and SAL1. So we are trying to use also the tetramers for try to purify these B cells, and try to use other protocols, not only tetramer, to sorting B cells. We just got in our institution a new flow cytome that Symphony That's a huge cytoma, many colors that can help in this. So to take home, these are very complex that I love. I got this in malaria vivax, they was virtual, but just to, so why you have to vote in DEC too? I hope that I have give so many evidence that yes, these are promised candidates. Of course, we not believe that only DEC will solve the problem of vivax, a very complicated parasite, but to have this elite responder that's present not only in the highly endemic areas such as Papua New Guinea, but also in the Amazon area where we have this fluctuation, malaria transmission. We have this. We saw that yes, there is okay. It's okay. There is this long memory B cell, so you can for vaccine development. This could be very important. So have to learn what they have difference. I also can see that have good and bad antibody response. So try to understand the good ones. So yes, we are confident about this. What we are doing now is try to characterize this B cell subpopulation, concentrating this B cell elite response. That's a task for now has been a lot of work for Maria Fernanda, that's postdoc for my lab, and Lucas Simões. They are have a hard work now, trying to set up all the conditions to try to understand this better. So I hope soon have results about this. Just to know you guys that beyond the DBP, this collaboration with the University of South Florida in Jones in France has been very, very good contribution, not only because we share protocols, improve our skills of our students, also the people from University of South Florida can come here, because we make a lot of contribution in parallel for science. I just emphasize too, for example, we have been start with work with Francis in, to try to characterize a novel erythrocyte bind protein that seems to be an alternative protein relevant for Plasmodium vivax invasion, perhaps in the bone marrow, because the reticulocyte that express this protein is more common in very young reticulocytes. That's more common in the bone marrow. And more recently, they started France. Now this Barbara, our student, so France helped us to try to characterize more the immune response here in the outbreak against this protein. Also, another hypothesis that to make, because in the Amazon area of Brazil, we have a lot of virus. So although Epstein-Barr virus, a common virus around of the world, but in the Amazon, the children, they sort of convert for Epstein-Barr virus much early. That, for example, in my region here in Brazil. Here in more developed regions of Brazil that have more income. So it's the teenager that change for seroconvert for Epstein-Barr virus. But in the Amazon, you saw that's very young children. We don't, it's different from what we have in Africa, that we have this associated cancer with falciparum, but yes, we don't have this in Brazil. So we make the hypothesis that perhaps this people that has this persistent detection of the DNA of the Epstein-Barr virus, because this virus go to the B cells forever, perhaps they could have, this can compromise the immune response of malaria. So we make just a proof of concept in study. And seems, yes, the answer is yes. We believe that this suggests that some degree of decrease in the immune response against different Plasmodium vivax proteins that are candidate of vaccine are decreased when this people has a persistent detection of virus. So another that I do like, so here, this is Gabi Assis, there's a student of our research for our group, Cristiana Brito. So yes, we try to use all these proteins in collaboration with John Adams and Francis to show that, to identify potential reservoir of malaria in our Atlantic forest. So we have these non-human primates, that Latin America, very small primate, non-human primates, that seems to be a zoonotic malaria. Sometimes we have outbreak of human infection, mainly for Plasmodium simio. That's a Plasmodium very similar to Plasmodium vivax. So yes, so we have a lot of contribution. Finally, I'd like to thank, this is John here in Brazil. You can see that a lot of students. I would like to thank Cristiana Brito and Flora. Thais is not here, that we are research in our group. We still have here Camila Valente. Now she's with Johnson Lab. She moves to there. And from the students that are here, I think that Luis is still one of our PhD students that go many years for the field. We have a lot of students. So, Franz, John, see, you have to come to Belo Horizonte sometimes. And yeah, I hope that John was back also, because the pandemic virus stopped all the trip. Just to illustrate also for you guys that in Brazil, different for USA, most of research in Brazil is made by undergraduate, master student, PhD student, and not postdoc. We have very few postdoc. So I think this profile is different that what's in USA, you have much more postdoc doing science. So I'd like to thank all the organizers and the support, especially the NIH, that this collaboration allowed us to go together with France and join this R01. So John was always very nice with us. I started my collaboration I was returned for a postdoc from NYU Medical Center. At that time I worked with just mice and CD8 T cells. I say, okay, I need to work with him. And I just write to John and say, okay, I know that we're doing a lot of DBP. Nobody's working with this here in Latin America. I do like that, if you can collaborate fast. And he said, 10 minutes later, yes. And we still since there. So thank you very much for John, for France, for all the other that stay in John's lab, and all the people from my lab, a lot of students that's not longer here, but have a lot of students out that keeping doing this, and all the collaborate from the field. I think this is the last. I cannot stop without saying this here. I have been working for many years with the Yanomami in Brazil, that people that live very isolated. So we come to know a lot of things about these people that when you start to get falciparum is not common in this region of Brazil, means they have gold miners in the area. So we have a lot of people die now in Yanomami. This is new for all newspaper around of the world, include science. So yes, these people should be safe of these illegal gold miners. So thank you very much. And I think that I'll stop to share my screen.
Okay, thank you very much, Dr. Carvalho, for that wonderful presentation. That was very insightful. I will confess that there's certain things that I've learned from this presentation which I've not known about despite the many years of collaboration with you. So ladies and gentlemen, I'll open the floor for questions.
Yes, I think that I have two here in the chat.
Yeah, there are some questions in this chat.
Yes, the first one is, let me check, I don't know if I can. The first one is, how do you form a relationship with the rural settlement in order to conduct your research? Usually we have this, we go we have a long way to talk with the people from the community. In this, for example, I'm talking about specific, because I have been working in the Amazon for 30 years, since I was a baby. No, yes, I make my PhD in the Amazon at that time as a student, so we have a lot of contact with the MK. So in Rio Pardo, is this community that we have been working. We start in 2008, we have this school. So usually we arrive there and talk with the people. Because Fiocruz is from the Brazilian Ministry of Health, and we have a lot of support. So the people usually know what Fiocruz doing in the field. So we are not only in the area, so we go there and give class about the disease. All the time that my students go, they go to play games in the school to learn how is malaria is transmitted. So they go, a lot of people there to teach children, the professor. So all the time we try to make this. And before we start and talk with the people, explain what's our project, what we're doing there. So you make this. So have a lot of contact, keep it. So when you arrive there, perhaps Flora is much more good to do this than me, because Flora stayed there for all these years. People, oh, are you back? If the people from Fiocruz. So usually, for example, in the COVID, they went to some tests. If we have people sick, we suggest we have car that we can send people for the city. We can improve this, yes. And sometimes we ask people to go, other people that work, for example, for leishmaniasis or fevers, that we can go together, but sometimes this is complicated. But for example, just for you know, when I started that time that we have a lot of falciparum in the Amazon, we go there. Many times we arrive, we have children in the house very sick, so we stop, take the children, go to the hospital. Fortunately, now in Brazil, and in Latin America, this Plasmodium vivax that predominates, that's not so kills so fast like falciparum. But yes, we provide some. But most what we do is like this, try to learn. So welcome. And there is another that how I can understand, how can I undergrad students at USF learn more information about this work. I think that you can write my email. I don't know if I can, I cannot have access to the chat, but Francis can.
Yes, I'll
can
put the email
put
address.
my email, yes, and you can contact us. So cannot be only malaria. I was talked before with the people from Linet, if I'm not wrong, Dr. Linet, and say, okay, you can make more workshop that for you people know more Fiocruz and try in US South Florida and make this connection. We received already one student, a graduate student, right, from Jones, I don't remember his name, but yes, we can try. We are now have an international support team. I'd like to make this more strong. I think this is a good idea. We can learn with you guys and you can learn with us. Okay?
Okay, I think we have some more questions from the chat. Would there be any interest in doing this study in India?
No,
yeah, absolutely. Maybe I can try to answer this. Yeah, absolutely. We are always looking for international collaboration. So we are open to expanding to other countries. And then there's also another question. Do we know if the allied responders also...
Oh, sorry. Now I saw the chat.
Do we know if the allied responders also have the similar IgM responses?
The point is, the elite responder, we only saw that they have this long immune response, but we don't know so many. I cannot answer this because we don't know. I think that because we have the long IgM response, not only for the elite responder, also for people has been long-term exposed to the area. So I think that the just peripheral antibody response will not answer this. So I don't know yet what's the difference, but I think that we have to go to check the B cells. So I don't know.
And then there's also so I just had a question about differences in DBP SAL1 and DEGNOL2. Why is it that SAL1 is able to achieve TH1, TH2 differentiation in the host while DEGNOL2 cannot?
No idea, but I'm so happy that there is difference between SAL1 and DEGNOL2. That means that we are in the not wrong way, because SAL1 go to this strain specific and not DEGNOL2. So, but that difference gene expression, I will not concentrate in that because it's can be everything. I showed this result some times ago for Lou Miller, the famous Lou Miller. So he told me, this could be a lot of things. Yes, I agree. This could be a lot of things. So I don't have idea, sorry. But I think that's just an open question that suggests us if there is difference between elite and non-elite, and difference in SAL1 and DEGNOL2, I think that's good. I will not interpret it like one go to stage 1, stage 2, but just because you saw this difference in gene expression, but this is, we have to make more controls to be sure about that. First question, first answer. And also, yes, we don't know. I think that we have to purify and check, because this was like this. We would like to see something like in a needle, in a lot of, in a different huge world of genes. So I don't know. I just like I saw that. Thanks, John. He said, for great historical perspective of our DBP story. Yes, and thank you very much, John, for your great collaboration. Okay, sorry, I don't know if I read, you read first.
So,
You can
yeah.
go.
So there's also Lynette Menendez that our office is happy to support further collaborations with Field Crews. Thank you for this excellent talk.
I put, just for people know, I put also the email for Samia Franco, that's from our international team. So include for the people from University of South Florida, if you'd like to contact more, and would like to make more workshops, not specific about malaria, but yes, and for also for students that perhaps are here in the audience that can write for some, and we can try to organize check.
Yeah, we still have some time for more questions, please. So a quick question here is why the elite responders and what make them so elite?
Excuse me, Francis, could you repeat?
So why the elite responders? Why do we have elite responders and what make them so elite?
This is a question of $1 million. And if you know this, we can go to a very good vaccine. I believe, France, they have difference in the long-term memory. And I also, but I'm telling you, we are so concentrated in the elite response, but they have a very complicated immune response because, I don't know, I would say, these people are not alone if familiar in the endemic area. For example, not only here in Brazil, but we have a lot of virus, a lot of other diseases that can make this immune response. But the good news is we know that part of this can be associated with clinical protection. But I'm check this data and say, okay, our preliminary data, you cannot see very difference between the B cells, between elite response and not elite response. So we are like this, we need to find something very different. But perhaps the people from the outbreak, we can learn more about the people from the outbreak because it was a single plasmodium virus infection and they have memory. So I think that perhaps our experimental approach should be try to understand the single infection with this elite responder, and not only elite versus the same area not respond. So I don't know, but I hope to see some difference in B cells because I don't know. So this is the idea. We have use now this try to use the point of the symphony that has a lot of colors, but we hope to go to the endemic area in return because we don't have so much frozen cells now. And also be back in that idea of John's that try to make the human monoclonal, perhaps we have to do C, because I think that we have very well characterized people, right? So we have to check this, what's this monoclone of these people see. And yes, I don't know what make them different, but they are different. And they are not different for example, MSP1, 19, because keep this forever, MSP1, 19, for everybody.
Mm-hmm.
So we have to answer this question together. Don't give up. You enjoy or not escape?
No escape. We're in for a long ride. So, yeah, I have a question here. It goes, what is responsible for the waning antibody titers in endemic regions?
What? Excuse me, could not understand.
What is responsible for the waning antibody titers? That for the reduction in antibody titers in endemic regions with time.
Why the antibody decrease?
Yeah.
Oh, because I think that conventional antibody decrease because they have short life, and probably for this, I don't know, oh, Francis. I have a lot of, I don't know, sorry, but this is supposed to be normal, this decrease, the wane. It's not normal, I think, it's not common, is that remain so high without malaria transmission. So I think that we only see this difference because we have very low transmission. And now I do like to know what happened now. I'm a little bit afraid because now we can see that we don't have very good transmission in that area. So yes, if you decrease transmission, decrease. We saw not for DBP, but for our other malaria, for example, MSP1, you can see if malaria increase antibody, decrease, make fluctuation. So it's because this the people try to see like biomarker of transmission, like Evo-Muli. So I think this is common, decrease, increase, but why they change for example, for the IgG increase for the DEC, no problem, because they have more immune protection. But why decrease for IgG for the SAL1? Because perhaps SAL1 is not circulate more when decrease transmission. So it's a lot of complex interaction between antibodies. But I think the big question, why this IgM is there?
Mm.
So, but the cycle of plasmodium virus is changed every day. So I don't know.
Okay.
But this is not only for malaria. I think this status, this IgM are just for virus. So yes, we cannot just concentrate only more in IgG.
Okay.
Open question.
Do we have any more questions from the audience?
Okay, we are on time. Just invite people to see this beautiful castle that's Fiocruz in Rio de Janeiro, and come to Brazil anytime, and Belo Horizonte. And yes, I think that Fiocruz has a lot of things to share with you guys.
Okay.
And now we have a new vaccine of COVID, that Brazilian vaccine against COVID, they are making a building here in Belo Horizonte to produce vaccine against different viruses and parasites.
Okay. Okay, so if there are no more questions, I would like to thank Dr. Carvalho for that wonderful presentation. I think it was very insightful, and I'm sure everybody learned a lot from this presentation. Thank you once more for the collaboration and all the work you're putting in through. I also want to give a big shout to Dr. Flora Cano, who was working with Dr. Carvalho. She's also been doing a very great job, especially in the field. And we very much appreciate what she's doing. There is Leticia, who was a PhD student who worked with us here at USF. Also, Dr. Cano was also here at USF for some time. And then now we have Dr. Camila Perez, who was working with Dr. Carvalho. She's now a postdoc here in the Adams Lab here at USF. She's doing a wonderful job and just got a paper accepted, which is in press. It's a great job. And we look forward to continue this collaboration and for more success in the future. Also want to throw a big thank you to Professor John Adams for all his support, and we'll forever appreciate his help. So thank you, everyone. Thank you for coming. We wish you all a wonderful day.
Thank you very much, Franz. Thank you, everybody. And Jessica Casanova that organized this. She's not here today, but thank you very much. And
yeah, very big shout also to Jesse and his team for
and Jamie that's here today.
Giving us this opportunity. Thanks.